Welcome back friends. Welcome back in another video tutorial for microbiology with Shakti. So today we will going to discuss a very important technique to detect amino acid, proteins and peptides. So that is the Ninhydrin test, the most commonly used technique, right? So this Ninhydrin test is based on the reagent Ninhydrin, which is also known as triketohydrinidine hydrate. So if I try to draw the structure of the Ninhydrin, so it somehow looks like that. There will be two OH and two ketone group. So this Ninhydrin thing will going to react with the alpha amino group of the amino acid. And alpha amino group is the amino group which is just beside the chiral carbon of the protein. Okay. So that is the idea. So this Ninhydrin will going to react with this alpha amino group. So first, this Ninhydrin, which is triketohydrinidine hydrate, is a strong oxidizing agent. And in presence of this Ninhydrin, this alpha amino group of the amino acid will be converted into ammonia, and liberation of CO2 aldehydes occurs and the Ninhydrin will be reduced. Okay, so let's directly move to the reaction scheme to understand it more clearly. So this is the Ninhydrin structure, this is the amino acids. So what will happen? Reaction between this alpha amino group and the Ninhydrin will occur, that will facilitate liberation of ammonia, aldehyde and carbon dioxide, and Ninhydrin will be reduced. Okay, so this step is oxidative deamination as well as decarboxylation. Okay, so as a result we will get reduced Ninhydrin, which we will call as hydrin dentin. Although this name is not necessary, you may just remember the reduced Ninhydrin. Okay, now what will happen, that this reduced Ninhydrin along with ammonia and another Ninhydrin which is in its oxidized form will going to react together. Okay, and as a result it will give you a blue colored complex, that is the Riemann purple, and it will give you a characteristics absorption at 570 nanometer. That will help you to determine the identity of the amino acid, as well as if you are analyzing protein, then determine the identity of the protein both quantitatively as well as qualitatively. Okay, so here the most important thing, that is, it will not only give you qualitative analysis as well as it will also provides you quantitative analysis, because the molar absorption coefficient for one amino acid, which may be simplest type of amino acid, that is glycine, should be different from the molar absorption coefficient for phenylalanine, right? So it may easily help you to understand the which kind of amino acid it is, or which type of amino acids are predominant in that particular protein. So that is the idea of Ninhydrin test. Okay, but it is quite different in case of proline, because if you study the structure of proline, there is no NH2, but instead there is a NH, right? Because proline's alpha amino group is involved in a reaction with the R group of the amino acid, right? So here NH is present instead of NH2. That is why we are calling proline as amino acid, right? So when we will going to perform Ninhydrin test with proline, we will going to get a yellow colored compound instead of this blue colored compound, and obviously it will going to give you absorbance at some different absorption. Okay, so that is the idea of our Ninhydrin test. Concept is the same. You will add the Ninhydrin reagent, then you will perform colorimetric analysis, right? That is the idea. So I hope this video will be helpful for you. Thank you.